Critical quality attributes, or CQAs, are one of the most important concepts in nanomedicine CMC, and also one of the most common sources of misunderstanding and regulatory risk and failure. Unlike small molecule drugs, where quality is largely defined by chemical identity and purity, nanomedicines are multi-component systems. Their clinical performance is driven not by a single parameter, but by a combination of interdependent attributes. Because of this, regulators expect a much deeper level of understanding and justification for CQAs in nanomedicine programs. One of the most fundamental CQAs in nanomedicine is particle size and size distribution. Particle size directly affects biodistribution, circulation time, cellular uptake, and clearance mechanisms. Nanoparticles within a narrow size range may preferentially accumulate in specific tissues, while slightly larger or smaller particles can behave very differently in vivo. Even modest shifts in average particle size or increases in size distribution width can alter pharmacokinetics and safety profiles. A broader size distribution increases the likelihood of off-target accumulation, variability in exposure, and unpredictable clinical outcomes. For this reason, regulators closely scrutinize how particle size is measured, controlled, and justified as a CQA. Surface properties represent another critical category of CQAs. Surface charge, often measured as zeta potential, plays a key role in colloidal stability and biological interactions. Surface charge influences aggregation behavior, protein adsorption, and interactions with cells and immune components. In biological environments, nanoparticles rapidly interact with plasma proteins, forming what is known as the protein corona. The composition of this corona is strongly influenced by surface chemistry and charge, and it can significantly affect biodistribution, cellular uptake, and immunogenicity. Small changes in formulation or processing conditions can alter surface properties and lead to biologically meaningful differences, even if particle size remains unchanged. Shape, internal structure, and surface features can influence cellular internalization pathways, release kinetics, and degradation behavior. While morphology may be less sensitive to minor process changes than size or surface charge, it still contributes to overall product performance and consistency. Beyond physical attributes, chemical and compositional CQAs are equally important. These include drug loading, encapsulation efficiency, purity, impurity profiles, and the presence of degradation products. Variability in composition can directly affect dose delivery, release characteristics, and stability over the product's shelf life. Stability itself is often considered a higher-level CQA that integrates multiple underlying attributes. Physical instability, such as aggregation or particle growth, and chemical instability, such as drug degradation or surface modification, can both compromise product quality. Stability studies therefore play a key role in confirming that CQAs remain within acceptable limits throughout storage and use. Manufacturing consistency is tightly linked to CQA control. As nanomedicine processes move from laboratory scale to pilot and commercial production, variability can be introduced through changes in mixing dynamics, shear forces, equipment geometry, and raw material quality. Processes that appear robust at small scale may behave very differently when scaled up. Without a clear understanding of which attributes are truly critical, manufacturing changes may unintentionally impact CQAs. These changes often go undetected until later stages of development, where correcting them becomes significantly more difficult, costly, and time-consuming. To effectively identify and control CQAs, a comprehensive analytical strategy is required. In nanomedicine development, CQAs are not analytical checkboxes or static lists. They are the foundation of a robust CMC strategy and a key determinant of program success.